11. Muscular system. With the exception of some smooth musdy tissue, the muscular system develops from the mesodermal germ layer and consists of skeletal, smooth, and cardiac muscle. Skeletal muscle is derived from paraxial mesoderm, which forms somites from the occipital to the sacral regions and somitomeres in the head. Smooth muscle differentiates from visceral splanchnic mesoderm surrounding the gut and its derivatives and from ectoderm, pupillary, mammary gland, and sweat gland muscles. Cardiac muscle is derived from visceral splanchnic mesoderm surrounding the heart tube. Striated skeletal musculature, head musculature is derived from 7 somitomeres, which are partially segmented whorls of mesenchymal cells derived from paraxial mesoderm. Musculature of the axial skeleton, body wall, and limbs is derived from somites, which initially form as somitomeres and extend from the occipital region to the tail bud. Immediately after segmentation, these somitomeres undergo a process of epithelization and form a ball of epithelial cells with a small cavity in the center, Fig.IB. The ventral region of each somite then becomes mesenchymal again and forms the sclerotome, Fig.IB, the bone-forming cells for the vertebrae and ribs. Cells in the upper region of the somite form the dermatome and 2 muscle-forming areas at the ventrolateral, VLL, and dorsomedial, DML, lips, or edges, respectively, Fig.IB. Cells from these 2 areas migrate and proliferate to form progenitor muscle cells ventral to the dermatome, thereby forming the dermal myotomy, Figs.1b and C2. Some cells from the VLL region also migrate into the adjacent parietal layer of the lateral plate mesoderm, Fig.IB. Here they form infrahyoid, abdominal wall, rectus abdominis, internal and external oblique, and transversus abdominis, and limb muscles. The remaining cells in the myotome form muscles of the back, shoulder girdle, and intercostal muscles, Table Point 1. Initially, there is a well-defined border between each somite and the parietal layer of lateral plate mesoderm called the lateral somitic frontier, Fig.IB. This frontier separates 2 mesodermal domains in the embryo, one the paraxial domain that comprises the region around the neural tube and contains only somite-derived, paraxial mesoderm, cells. 2 the abaxial domain that consists of the parietal layer of lateral plate mesoderm together with somite cells that have migrated across the lateral somitic frontier. Muscle cells that cross this frontier, those from the VLL edge of the myotome, and enter the lateral plate mesoderm comprise the abaxial muscle cell precursors and receive many of their signals for differentiation from lateral plate mesoderm, Fig.3, those that remain in the paraxial mesoderm and do not cross the frontier, the remaining VLL cells and all of the DML cells, comprise the paraxial muscle cell precursors and receive many of their developmental signals from the neural tube and notochord, Fig. 11.3. Regardless of their domain, each myotome receives its innervation from spinal nerves derived from the same segment as the muscle cells. The lateral somitic frontier also defines the border between dermis derived from derm atoms in the back and dermis derived from lateral plate mesoderm in the body wall. It also defines a border for rib development, such that the bony components of each rib are derived from paraxial sclerotome cells and the cartilaginous parts of those ribs that attach to the sternum are derived from sclerotome cells that migrate across the lateral somitic frontier, abaxial cells. Innervation of axial skeletal muscles, the new description of muscle development characterized by paraxial and abaxial domains differs from the oid concept of epimeres, back muscles, and hypomeres, limb and body wall muscles, which was based on a functional definition of innervation. Epimeric muscles were innervated by dorsal primary rami, hypomeric muscles by ventral primary rami. The new description is based on the actual embryological origin of muscle cells from 2 different populations of muscle cell precursors, the abaxial and paraxial cells, and not their innervation. The description does not preclude the fact that epaxial, above the axis, muscles, back muscles, are innervated by dorsal primary rami, whereas hypaxial, below the axis, muscles, body wall and limb muscles, are innervated by ventral primary rami, Fig. 4. Skeletal muscle and tendons. During differentiation, precursor cells, the myoblasts, fuse and form long, multinucleated muscle fibers. Myofibrils soon appear in the cytoplasm, and by the end of the third month, cross striations, typical of skeletal muscle, appear. A similar process occurs in the 7 somitomeres in the head region rostral to the occipital somites. However, somitomeres never segregate into recognizable regions of sclerotome and dermal myotomy segments prior to differentiation. Tendons for the attachment of muscles to bones are derived from sclerotome cells lying adjacent to myotomes at the anterior and posterior borders of somites. The transcription factor scleraxis regulates development of tendons. Molecular regulation of muscle development. Genes regulating muscle development have recently been identified. Bone morphogenetic protein 4, BMP4, and probably fibroblast growth factors from lateral P8 mesoderm, together with WNT proteins from adjacent ectoderm, signal VLL cells of the dermal myotomy to express the muscle-specific gene MyoD, Fig.3. BMP4 secreted by ectoderm cells induces production of WNT proteins by the dorsal neural tube at the same time that low concentrations of sonic hedgehog, SHH, proteins, secreted by the notochord and floor plate of the neural tube, reach the DML cells of the dermal myotomy. Together these proteins induce expression of MYF5 and MyoD in these cells. Note that SHH does not play a role in specifying VLL cells. Both MyoD and MYF5 are members of a family of transcription factors called myogenic regulatory factors, MRFs, and this group of genes activates pathways for muscle development. Patterning of muscles. Patterns of muscle formation are controlled by connective tissue into which myoblasts migrate. In the head region, these connective tissues are derived from neural crest cells. In cervical and occipital regions, they differentiate from somitic mesoderm, and in the body wall and limbs, they originate from the parietal layer of lateral plate mesoderm. Head musculature. All voluntary muscles of the head region are derived from paraxial mesoderm, somitomeres and somites, including musculature of the tongue, eye, except that of the iris, which is derived from optic cup ectoderm, and that associated with the pharyngeal, visceral, arches, table 02, Fig. 2. Patterns of muscle formation in the head are directed by connective tissue elements derived from neural crest cells. Limb musculature. The first indication of limb musculature is observed in the 7th week of development as a condensation of mesenchyme near the base of the limb buds, Fig. 2. The mesenchyme is derived from dorsolateral cells of the somites that migrate into the limb bud to form the muscles. As in other regions, connective tissue dictates the pattern of muscle formation, and this tissue is derived from the parietal layer of lateral P8 mesoderm, which also gives rise to the bones of the limb. Cardiac muscle. Cardiac muscle develops from splanchnic mesoderm surrounding the endothelial heart tube. Myoblasts adhere to one another by special attachments that later develop into intercalated discs. Myofibrils develop as in skeletal muscle, but myoblasts do not fuse. During later development, a few special bundles of muscle cells with irregularly distributed myofibrils become visible. These bundles, Purkinje fibers, form the conducting system of the heart. Smooth muscle for the dorsal aorta and large arteries is derived from lateral plate mesoderm and neural crest cells. In the coronary arteries, smooth muscle originates from proepicardial cells and neural crest cells, proximal segments. Smooth muscle in the wall of the gut and gut derivatives is derived from the splanchnic layer of lateral plate mesoderm that surrounds these structures. Only the sphincter and dilator muscles of the pupil and muscle tissue in the mammary and sweat glands are derived from ectoderm. Serum response factor, SRF, is a transcription factor responsible for smooth muscle cell differentiation. This factor is upregulated by growth factors through kinase phosphorylation pathways. Myocardin and myocardin-related transcription factors, MRTFs, then act as coactivators to enhance the activity of SRF, thereby initiating the genetic cascade responsible for smooth muscle development. Clinical correlates. partial or complete absence of a muscle is common and usually not debilitating. Examples include partial or complete absence of the palmaris longus, serratus anterior, or quadratus femoris muscles. AM or serious defect is called Poland sequence THAT, occurs in 1 per 20,000 individuals and is characterized by absence of the pectoralis minor and partial loss of the pectoralis major, usually the sternal head, muscles Fig.IB. The nipple and areola are absent or displaced, and there are often digital defects, syndactylly fused digits and brachydactyly short digits on the affected side. The disfiguring nature of the defects can be problematic, especially in females because of breast development. Partial or complete absence of abdominal musculature is called prune belly syndrome Fig.6. Usually, the abdominal wall is so thin that organs are visible and easily palpated. This defect is sometimes associated with malformations of the urinary tract and bladder, including urethral obstruction. These defects cause an accumulation of fluid that distends the abdomen, resulting in atrophy of the abdominal muscles. Muscular dystrophy is the term for a group of inherited muscle diseases that cause progressive muscular wasting and weakness. There are a large number of these types of diseases, of which Duchenne muscular dystrophy, DMD, is the most common, 1 per 4,000 male births. The disease is inherited as X-linked recessive, such that males are much more often affected than females. Both DMD and Becker muscular dystrophy, BMD, are caused by mutations in the gene for dystrophin on the X chromosome. However, in DMD, no functional dystrophin is made and the disease is much more severe with an earlier onset, 5 years of age versus 8 to 25 years of age in BMD. Dystrophin is a cytoplasmic protein that forms a dystrophin-associated protein complex linking the cytoskeleton to the extracellular matrix. Summary: most muscles arise from the mesoderm. Skeletal muscles are derived from paraxial mesoderm, including 1. Somites, which give rise to muscles of the axial skeleton, body wall, and limbs. 2. Somitomeres, which give rise to muscles of the head. Progenitor cells for muscle tissues are derived from the ventrolateral, VLL, and dorsomedial, DML, edges, lips, the prospective dermal myotomy. Cells from both regions contribute to formation of the myotome. Some cells from the VLL also migrate across the lateral somitic frontier into the parietal layer of the lateral plate mesoderm. This frontier or border separates two mesodermal domains in the embryo: 1. The paraxial domain that surrounds the neural tube and contains only somite-derived cells, paraxial mesoderm. 2. The abaxial domain that consists of the parietal layer of lateral plate mesoderm in combination with somite-derived cells that migrate across the frontier into this region, Fig.1. Abaxial muscle precursor cells differentiate into infrahyoid, abdominal wall, rectus abdominis, external and internal obliques, transversus abdominis, and limb muscles. Paraxial muscle precursor cells form muscles of the back, some muscles of the shoulder girdle, and intercostal muscles, Table 1. Muscles of the back, epaxial muscles, are innervated by dorsal primary rami. Muscles of the limbs and body wall, hypaxial muscles, are innervated by ventral primary rami. Molecular signals for muscle cell induction arise from tissues adjacent to prospective muscle cells. Thus, signals from lateral plate mesoderm, BMPs, and overlying ectoderm, WNTs, induce VLL cells, whereas signals from the neural tube and notochord, SHH and WNTs, induce DML cells. Connective tissue derived from somites, parietal mesoderm, and neural crest, head region, provides a template for establishment of muscle patterns. Most smooth muscles and cardiac muscle fibers are derived from splanchnic mesoderm. Smooth muscles of the pupil, mammary gland, and sweat glands differentiate from ectoderm.